In this slide, I'm showing you the model for anterior-posterior pattern formation according to the expression of different maternal genes. For example, you see in the oocyte, we have different messenger RNAs like bicoid, messenger RNA like Nanos and Oscar as well. And we have hunchback and caudal, two maternal messenger RNAs, which are ubiquitously expressed all over the anterior-posterior axis of the oocyte. We don't have any modification according to the anterior-posterior axis regarding these messenger RNAs in the oocyte. But after fertilization of the oocyte, if we look at these different gene expression in the embryo, so here we are looking at the proteins because these genes can be expressed at the level of the protein after fertilization. We see that bicoid, okay, and different other proteins, they can establish kind of gradients, okay, they diffuse and they establish gradient. The highest level of expression of bicoid is anterior. The highest level of Nanos is posterior. You see, this is the posterior, this is the anterior. And hunchback, okay, is also having high levels anteriorly, while caudal is having high level posteriorly. So how this is controlled? For example, here in the oocyte, bicoid messenger RNA, okay, it will be translated into bicoid protein in the embryo. And this bicoid protein here, it inhibits caudal in black, okay, it inhibits caudal messenger RNA from being translated into caudal protein right here anteriorly, okay? So we don't have caudal expression anteriorly because it's inhibited by bicoid. While posteriorly, we see that we have Nanos messenger RNA translated into Nanos protein right here, and as well for Oscar as well, so it's also expressed posteriorly. And Nanos and Oscar here, they inhibit, okay, hunchback in green from being translated into hunchback protein. So we don't have hunchback expression posteriorly, we have caudal expression instead. So what does it mean? So bicoid is an inhibitor of caudal. You see, we don't have caudal expression anteriorly because it's inhibited by bicoid. And caudal protein, you see, it's expressed posteriorly because it's important for the specification of the posterior domain of the Drosophila embryo. Okay, it will determine the caudal part, okay, or the posterior part. So they also found that one main target of bicoid, okay, is hunchback, okay. One of the main target genes of bicoid is hunchback, which is expressed anteriorly. So when they try to mutate, okay, or to make mutants which are deficient for hunchback expression maternally and the zygotic hunchback, because hunchback is expressed in the oocyte and later in embryo. When they made mutation for hunchback, they found that the embryo will lack the mouth part and the thoracic parts. It means they lack the anterior domain. Okay, so it means that maternal and zygotic hunchback contribute to the formation of the anterior pattern or the anterior compartment of Drosophila embryo. Okay, so it means like bicoid, but bicoid is more important than hunchback for the formation of the anterior compartment. Okay, so because bicoid control hunchback expression, okay, and not the opposite. So we already talked about this part, okay, from the head to the abdomen. So it means the segmented part of the Drosophila embryo, okay, that includes the head, the thorax, and abdomen. All this part is segmented. We have 3 head parts, 3 head segments, 3 thoracic segments, and 8 abdominal segments. But we also have in Drosophila embryo unsegmented extremities. So these unsegmented extremities are called acron anteriorly and telson posteriorly. So the telson includes the tail and the acron includes the head or the brain. So what kind of genes control acron and telson? We have a gene called torso. Torso, it's a gene encoding an RTK, receptor tyrosine kinase. Okay, so the mothers having mutations for torso cannot develop neither acron nor telson. So it seems like torso is very important for the determination of acron and telson. So at which stage torso start to be expressed? So it seems like if we look earlier in the oocyte, we can already see torso expression. Okay, you see this peripheral part of the oocyte, okay, in pink color, okay, around the yellow you see here in the periphery. Okay, this is the torso expression. Okay, it's evenly distributed throughout the plasma membrane of the oocyte. But usually acron and telson cannot be developed everywhere. They are developed acron anteriorly and telson posteriorly. So how acron and telson can be formed? It seems like torso is only activated in this location and in this location at the anterior and the posterior domains by another protein very similar to torso, expressed by the follicle cells of the anterior part and the posterior follicle cells of the posterior part. And these follicle cells express a gene called torso-like. Okay, torso-like. And this torso-like, when it's secreted by the follicle cells, it can pass through the perivitelline membrane and enter the cytoplasm of the anterior and the posterior sides of the oocyte in order to activate torso only in these locations. Okay, so that acron can be formed right here and torso right here at the opposite domain. So it seems like if we make mutation in torso-like or in torso, we get the same phenotype. It means we don't have any formation of acron and telson in this case. Okay, so torso is only activated by torso-like. So here they try to make one experiment by it's a gain of function mutation of torso. Gain of function mutation, it means they induce a constitutive activation of torso. Okay, they induce a constitutive activation of torso, and this was able, okay, the activation of torso was able to convert the anterior half into acron and the posterior half into telson. It means if we activate torso right here and torso right here, we are able to make acron and telson. And let's say we injected torso-like in these follicle cells right at this location. Okay, torso-like can diffuse through the perivitelline space and enter the membrane or the cytoplasm of the oocyte at this location in order to activate torso right here, and it will be able to create acron or telson. Okay, so in this slide you can see that torso gene are expressed only in these unsegmented regions. Okay, right here anteriorly and right here posteriorly we have torso. Okay, but in the middle we don't have, so in the segmented part we don't have any torso expression. Okay, only anteriorly and posteriorly we have torso, but in the middle we have a Gaucher protein. This Gaucher protein expressed in this domain is able to inhibit torso expression in the middle. Okay, it's able to inhibit torso expression, so we don't have any acron or telson between anterior and posterior extremities of the embryo. Okay, you only need to know this information from this slide. You don't have to go through talus and hook vein. Okay, just this is not required. You only have to know this information, what I explained right here on the graph. So if we summarize what we explained until now in this chapter, we have seen genes that define anterior organizing center. What is the anterior organizing center? They are the genes that will define the anterior domain, like bicoid, for example, that will activate the gap genes. And usually in the anterior domain, the bicoid and the gap or the anterior determinants like bicoid will repress the expression of posterior specific gap genes anteriorly. It will only allow the anterior gap genes to be expressed. We also talked about posterior genes that will form a posterior organizing center. For example, Nanos and Oscar are posterior genes. They inhibit the formation of anterior domain, and they inhibit the expression of anterior genes posteriorly. Okay, and we finally talked about the genes that determine the terminal extremities or the terminal boundary regions, like for example acron and telson, and we have seen that torso is an essential gene for the formation of acron and telson.